Hi guys, today we're going to take a look at chapter 1. Chapter 1 is just an introduction into microbiology, so kind of a short and sweet chapter, but I just want to kind of introduce what microbiology means, the definition itself, what we're studying in this class, and what we're going to be taking a look at throughout the next 10 weeks. So this chapter is a great way to introduce that to you guys. So in your book, we are looking at chapter 1 if you're following along with us today. If not, no big deal. We've got the PowerPoint that I'm going to share with you right now. So here is the PowerPoint. So I'm going to go ahead and get this started. Well, let me move this over here. Okay, here we go. All right, guys. So from the beginning here, let's take a look. So again, we're looking at an introduction into microbiology, the science itself. So let's talk about what that is. So in this chapter, we are looking at just that, what microbiology is and why we study it. I think I may have taken this out, some of the first microorganisms on Earth. If we didn't, we'll briefly touch on that. And some of the same thing with the earliest known infectious diseases. We're actually going to touch on that a little bit later in the course. We are going to definitely take a look at some of our pioneers in the science of microbiology. There's 3 individuals that we are going to take a look at, and we're going to see why they are important to the study of microbiology. And then this chapter typically finishes out with careers in microbiology, which I typically skip over because you guys have already decided on a career. You guys have picked nursing. And I'm just going to kind of end the chapter and touch a little bit on why microbiology is going to be important for you guys. So let's take a look at this. Okay, so biology itself, if we were to look at biology just by itself individually, if you were just taking a plain simple biology course, you are studying living organisms. That's what that is. Ology, anytime you see ology, you're looking at the study of something. So any type of ology class that you guys have seen or taken, you're talking about the study of something. But that prefix gives you a little hint as to what that is. And bio means life. So biology is the study of living things. So microbiology is going to be a little bit more of an advanced course to that because we're looking at small life forms. But even more advanced than that, not everything we're going to study in this chapter, I'm sorry, in this book or in this course is a living thing. In fact, next week, we're going to take a look at the study of viruses. And we're going to talk about how viruses are actually not living at all. Viruses are very, they're really kind of just small particles. They're sometimes just called non-living entities, but they actually are very dependent on the host, on what it is that they are infecting in order for them to be able to spread. They use the host living cells in order to then colonize and spread. So it's not necessarily, when we look at if we were to break down the word for those of you guys who have had medical terminology, and that's what you're tempted to do right here, and you're looking at micro, meaning small or tiny, bio, meaning life, ology, meaning study, you guys are thinking, okay, well, small life or small living organisms. Instead, we are talking about small, but that's going to include both living and non-living entities. And that's what it tells us here on the next bullet point there. Microbiology is the study of microbes, which is just a short way of saying microorganisms. So anytime you hear me say microbes, I'm just shortening microorganisms. So microbiology is the study of microorganisms, which are extremely small. So meaning that that's where that word micro comes from. For those of you guys who had, I know math with me, you guys should remember. For those of you guys who just had math with maybe somebody else, that's okay. But maybe you remember studying the metric system, and maybe remembering the prefix micro was the smallest unit that we looked at. So when we were talking about micrograms or microliters, then we're looking at something very, very small. Micro means that we cannot see these organisms with the naked eye. I can't just look at the table here in front of me and see a microorganism. I would need a microscope in order to be able to see that. And we're going to take a look at microscopes and how they work in the next chapter, in chapter 2 this week. So that's what they mean, that they're small, meaning microscopic. They're living, and then as I mentioned before, and certain non-living entities, and that's going to include our viruses and virons and prions, and that's all included in the category of viruses that we'll see next class. So that's what microbiology is. Essentially, just in a nutshell, if we were to just break it down, we're talking about the study of microorganisms. So that does include, again, viruses, as I've mentioned many times, but we're also going to take a look this quarter, we're going to take a look at bacteria. We're going to take a look at fungi. There are a lot of other microorganisms. In fact, if you were to have already skimmed through your book, you may have seen algae, archaea, protozoa. There's a lot of different microorganisms. But the reason why I simply just focus on, for you guys, especially as nurses, on viruses, bacteria, and fungi, those are the three most common ones that you guys will come across in the nursing field because of how they cause infections. Now, yes, others can, but we're going to take a look at those as those are your common ones that you guys will see. You guys are typically going to see viral infections, bacterial infections, and fungal infections in your line of work. So that's what we focus on in this term. So those are going to be what I mean when I say microorganisms. Those are probably all things you've heard of before. So that's what we're studying when we take a look at microbiology. So living organisms, or living microorganisms, I should say, can either be categorized as either cellular or acellular. And what that means, and hopefully you guys maybe remember this from anatomy, but anything that contains cells means it's living. In order for an organism to be a living thing, it has to be made up of cells. Human beings, and that's why I mentioned anatomy, human beings are living things, correct? So therefore, we are made up of cells. And you know that we have all kinds of different cells in our body. We have muscle cells, nerve cells, blood cells. We have just a ton. We are living. We're a living thing. We are considered a cellular organism. Plants and animals are both made up of cells, so therefore they are living organisms. But when we're talking about our microorganisms, especially the three common ones that we've mentioned, the most common ones, bacteria and fungi, are cellular because they're living things. Some bacteria just have one cell, but that one cell is all they need to make them a living thing. So they are cellular. So cellular is a synonym for living, if you think about that, putting those together. Whereas acellular, you guys can see where we're going, that A meaning without. So without cells is a synonym for non-living. Okay, so that's what we mentioned in that slide, or in that bullet point there. Living microbes are known as cellular microbes, and those examples would include your bacteria, archaea, some algae, protozoa, and some fungi. Whereas then your non-living microbes, therefore they are known as acellular, again, thinking without cells, meaning that they are non-living. So they are going to include, and then all these terms here we're going to talk about next class, viruses, but that also includes then viroids and prions, which are simply just smaller versions of viruses. We'll learn that next week. Microorganisms are, and it mentions in that last bullet point there, ubiquitous. What does ubiquitous mean? Ubiquitous means that they are found everywhere. And when I say everywhere, I mean really everywhere. They are in our atmosphere, they are on our desks in front of us, they are on our skin, in our bodies. Microorganisms are all over the place. And you guys are thinking right now, ew, I don't want microorganisms everywhere. But actually, the majority of our microorganisms in our life are actually really good for us. If we were to just looking at numbers, for those of you guys who had me in math, you guys know I like numbers. Well, as far as if we were to look at all of them, about 3%, only about 3% are microorganisms that are bad for us, meaning that they cause infections and cause us to get sick. That means 97% of our microorganisms are actually good for us. And that's what we're going to talk about a little bit in this chapter. I'm going to talk to you guys about a lot of different examples where microorganisms are used for good in our world. So let's take a look. So just as a reminder here, this is just a breakdown as what I've mentioned previously, that microbes can either be categorized as acellular, again, including those viruses because they're non-living, or cellular. Now, cellular, this is something that will be brought up again a little bit later. But when I say cellular organisms can be either categorized into what's called prokaryotes or eukaryotes. So remember, this whole section right here, cellular organisms, we're looking at they contain cells, therefore they're living. So we've already kind of excluded the acellular ones. But if we are just to break down those cellular ones, they are either categorized as prokaryotic or eukaryotic. And what does that mean? In short and sweet, a eukaryotic microorganism, or even a regular organism like a human beings would fall into the eukaryotic category because they contain membrane-bound organelles. What does that mean? What that means is in a cell, so I'm going to draw a picture here just to kind of give you guys an idea. So if we were to say that this circle, if you guys can see my screen up there in the corner, if we were to say, in fact, let me just stop sharing for a second. If we were to say that this circle is a cell, okay, again, we're talking about cellular organisms, then if for anybody who's ever seen a cell, I know we didn't really dive into a cell in anatomy for those of you guys who had my anatomy class. Any cell has lots of different parts in a cell. So if you guys are familiar with terms like the nucleus or the Golgi body, mitochondria, which gives cells its energy, ribosomes, which are proteins for your cells, you know, there's a lot of different parts. And all of those parts are kind of contained on their own. So for instance, I'm going to tell you guys the nucleus. Any cell has genetic information in it, and that tells it what its job is. So all cells have some type of DNA or RNA, which is their genetic info. Like for instance, in a neuron, which is a nerve cell, there's a nucleus in the middle of it. So there's that circle that has all that DNA in it. I'm going to try to draw. Oh, that's horrible. There's my double helixes there. Those are terrible, but those are that's that genetic information. It's housed in that nucleus. So that's just an example of a part of a cell, but notice that that nucleus has this wall around it. That's a membrane. That membrane is keeping that DNA in one place. That's what a eukaryotic cell is. It has these membrane bound, and when I say organelles, I mean it's different parts. So that there's other parts besides just a nucleus, but all of them are kind of contained in their own little section in a cell because they're surrounded by this membrane that keeps it in place. Whereas a prokaryotic cell, so let's call this a prokaryotic cell, instead, that genetic information that's normally housed in that nucleus is just free floating all around the cell because there's no nucleus keeping it in place. It just kind of floats wherever because there are no membrane-bound organelles. So that's the difference. So this is your eukaryotic cell, that E, and this is a prokaryotic cell. So that's a big difference between the two. That again, eukaryotic cells have these membrane-bound organelles in it, so different pieces that are kind of in their own section, and prokaryotes do not. So let's go back to that. All right, hold on, sorry guys. All right, let's go back to that slide there. Here we go. So that's the difference. So as far as the microorganisms that we're going to study, eukaryotes are going to be the fungi that we're going to talk about when we cover chapter 9. Whereas then prokaryotes, we'll talk about bacteria in a couple weeks, and that we'll see those are those prokaryotic cells. So that one that had that DNA free floating, bacteria just let that DNA float throughout the cell. That's what it does. So those are the difference between your two types of cellular organisms. And again, we'll bring that up a little bit later too. Okay, so why do we study? First, again, we're still talking about what is microbiology. Remember on not the recent slide, but the slide before, I talked about how microbes are ubiquitous. Microorganisms can be found virtually everywhere, but only about 3% of them are bad for us. And those are what we commonly refer to as germs. So the microbes that cause disease, those are sometimes referred to as germs, but the more scientific term for that, those disease-causing microbes, are going to be called pathogens. You all may or may not have heard the term pathogens. Pathogens is very similar to the term antigen. If you guys maybe remember the term antigen from microbiology. You're in microbiology, from anatomy. Antigen meant something foreign. But when I say the term pathogen, it doesn't just, fall into something foreign. A pathogen is specifically referring to disease-causing microorganisms. So this would be only, bacteria, viruses, things that cause diseases in our body. Those are called pathogens. This would not include things like, because when I say the term antigen, antigen could also include a tumor. An antigen could also include, like, if type B blood, somebody who's type B was given type A, the A antigen would be considered something foreign in that body. So again, that would be considered an antigen, whereas those are not microorganisms, for example. So just a heads up on that. So pathogen is specifically is talking about a disease-causing microorganism. Whereas then, therefore, microorganisms that do not cause disease, those are going to be referred to as non-pathogens. And remember, as I mentioned earlier, these are going to be the vast majority of our microbes. As again, I mentioned that about 3%, only 3% are bad for us, are going to actually cause diseases. So only 3% of all of the microorganisms in our world are pathogens, meaning that the majority of them, about 97% of them, are non-pathogens. They're good for us. And let's see where we see some examples of this. So first, when we're talking about the human body, the human being, microbes live in and on our bodies. We've got microbes that live on our skin. We've got microbes that live inside of our body, in our digestive tract, for example. These are referred to as indigenous microflora. Some people may refer to this as microflora, or if you've ever seen sometimes like probiotic commercials or yogurt, oftentimes say we need to restore our body's natural flora. That's what it's doing. It's shortening this. This is the term for that. Indigenous microflora, indigenous meaning it belongs at that particular habitat. And our microflora, that's, again, that habitat is our body. So it's our microorganisms that are good for us, that live in and on our body. We'll bring up indigenous microflora quite often as we go through different chapters, even when we talk about how our bodies protect itself against the pathogenic, microorganisms, the ones that are bad for us. Sometimes it's this indigenous microflora that are going to help to fight that off, because they're the ones that normally live on our body. So they're going to make sure that they stay and the bad ones leave. So indigenous microflora, again, are good for us. Some members, however, of this indigenous microflora are called opportunistic pathogens. What an opportunistic pathogen is, as it mentions in that next bullet point there, they're microbes that can cause disease, but sometimes don't. I shouldn't say sometimes, they usually do not. They can be thought of as microbes that are really just kind of awaiting the opportunity to cause disease. Well, what would that opportunity be? Well, let's talk about a couple different examples. One, sometimes when our immune system is weakened for whatever reason, if maybe somebody's going through chemotherapy or radiation, or they're just, sometimes pregnant women have a weakened immune system. So for whatever reason, sometimes when our immune system kind of goes, I'm not going to work as well as I normally do, sometimes that indigenous microflora can therefore take that as that opportunity. But a great example, I think, would be this. One of our indigenous microflora that lives in our body would be, an example would be like E. coli. Now, there's different forms of E. coli. Sometimes you can get bad E. coli from eating undercooked burgers or meat, and that can make you really sick. But there is a type of E. coli that lives in our digestive tract that actually helps to digest food as it's coming to. So it's a bacteria that lives in our gut that helps in that process of digestion. So it's very good for us. That is an indigenous microflora. It lives there normally, actually helps a process that our body normally goes through, and make sure that that happens as it should. But sometimes in the event that an indigenous microflora leaves the anatomical site that it normally lives in and goes into another anatomical site in our body, it doesn't recognize where it is, and then therefore can cause an infection. I use the example E. coli because, especially in females, Because if E. coli leaves the digestive tract, for instance, in a situation where maybe a female is using the restroom and happens to wipe from back to front instead, sometimes that can take E. coli from the end of our digestive tract, from the anus, and move it to the urethra. And then therefore the urethra, again, is the end of the urinary tract, but that can cause E. coli to go up into the urethra or up into the bladder. And actually, E. coli is the #1 cause of urinary tract infections. So therefore, that was given the correct opportunity. So sometimes that's the opportunity, that the microflora leaves the anatomical site where it's normally supposed to be, and it finds itself, like in this case, we're talking about the digestive tract moving to the urinary tract, and now it goes, I don't know where I am, and it causes an infection. So that's the opportunity, and that's what I mean by that. So that's what I mean by the term opportunistic pathogen. So when pathogens do cause disease, they cause one of two categories of diseases. They can cause either an infectious disease or a microbial intoxication. And so here's what this means. Infectious disease, what this means is the pathogen has made its way into our body. What it does is that it then colonizes, or it spreads within the person's body. So an example would be like if, I don't know, let's say somebody who has the flu sneezes, goes and opens the door. I go behind them and open the door, and then I go and wipe my nose. I may then take that pathogen and it makes its way now into my nose. That pathogen is now going to spread throughout the person's body, or through my body. I'm using me as an example. And the pathogen itself causes that disease. This is known as, again, an infectious disease. In a microbial intoxication, what happens here, the pathogen actually produces its own toxin. And that toxin is what is taken in by the person's body, and it's the toxin itself that causes the disease. So in an example where somebody maybe ingests a bacteria, or the bacteria that's producing this toxin or exotoxin, that toxin itself is what causes the disease. So that's called a microbial intoxication. So there's the difference, that when a pathogen gets into the body and it itself is what spreads and colonizes throughout the person's body, that's called an infectious disease, which we'll talk a little bit more about a little bit later. And a microbial intoxication, the person has ingested the toxin that the pathogen has created. Both very, very similar, as again, both make an individual very sick. So why do we study microbiology? So again, as I mentioned, these ubiquitous microorganisms that are found everywhere, we're going to take a look at some examples of why they're good for us. So microorganisms play very significant roles in our lives, besides the indigenous microflora that we just talked about. But there are also microorganisms that are very essential to the planet, for life on the planet. They play very important roles in the elemental cycles in our atmosphere. For instance, photosynthetic algae, as we know, plants go through the process of photosynthesis. If you guys aren't too familiar with the term photosynthesis, trees and flowers and plants, by taking in sunlight and soil, they will grow, and at the same time then can produce oxygen for our atmosphere. So the trees themselves are what is helping to provide oxygen for our atmosphere. Well, we have algae that also happens to be photosynthetic, and we have some photosynthetic bacteria as well. An example of that would be what's called cyanobacteria. These particular types of microorganisms are able to go through that process of photosynthesis and therefore help to produce more oxygen in our atmosphere. So they help the same way the trees are doing. We also have microorganisms that are involved in the decomposition process of any dead organisms or waste products that are, on sides of the road or in forests. If something dies, if a tree falls over and starts to rot, then what will happen is that rotting process. What takes place is some microorganisms are helping to eat that matter, help to eat that dying matter. And then what it does is it provides nutrients for our soil and then therefore helps to, provide those nutrients for our plants and helps in our other elemental cycles. An example of one of these decomposers would be an organism called what they're called as saprophytes. Saprophytes are organisms that live on that dead or decaying organic matter. And that's what they're doing, is they're helping to break down and decompose those organisms and that organic matter. Organisms, our microbes, are sometimes used to clean up toxic wastes and other industrial waste products. This is known as bioremediation. In the event that sometimes there's an example in a video I watched, and I'll have to just explain it, but there was a town that was a mining town, and in the mining, industry, what it does, it was producing a chemical called cyanide, and it was putting it into the town's water supply. And that was very bad. We don't want cyanide in our water supply. It was killing off all the fish, and if people drank that, it could be very harmful to them as well. So what they did, they could have either spent millions of dollars on a chemical that helped to counteract the cyanide, or what they did was they used more of a biological process, and they used microorganisms that are particularly good at cleaning up this. They're able to live in water, and I'm going to talk to you guys about bacteria in a couple weeks on how there's a difference between bacteria that can live in an oxygen-filled atmosphere like the one we're in right now. But there are some bacteria that are called anaerobes that can live without oxygen. So they're able to live in the water, and in that process, they're able to clean up that cyanide. So that's just an example of what's called bioremediation, where sometimes, and that was a much cheaper process for these individuals in this mining town. This was just an example. Like I said, I saw it in a video once, and about how microorganisms help to clean up our atmosphere as well. So that's an example of where, again, we're seeing good microorganisms. So here's an example of those saprophytes that I was mentioning. Like I said, if a tree falls over and a log starts to rot, it doesn't have its roots anymore. It's not getting the nutrients that it needed. It's going to die. But as it does, those saprophytes are going to help to decompose that log and help to provide nutrients for our soil, as you can see here. Many microbes, as I've mentioned, play essential roles in various elemental cycles. So I've mentioned the oxygen cycle, but there's a lot of other elements in our atmosphere, in the air, besides just oxygen. There's also carbon and nitrogen, sulfur, phosphorus, and microbes help to play a role in this as well by providing those nutrients to the soil. And then in that process of getting those elements into our air, that the microbes help to play those essential roles in that process. Algae and bacteria are also food. They're food for other organisms, such as tiny animals. So therefore, they're important links in our food chain, because then again, remember the food chain, then, larger animals can eat the smaller animals and provide food for, even humans. So they're important links in the food chain. Microbes that live in the intestinal tracts of animals, as we've mentioned with E. coli, for example, as I talked about with that indigenous microflora, microbes are good for that process as well. So living in the intestinal tracts of animals and humans help to aid in the digestion of food and help to produce beneficial substances. Microorganisms are also used in science. Science is, really just, I know microbiology is a scientific study, just looking specifically at microorganisms, but really all science itself is really the process of questioning things and studying and asking ourselves, how can we improve this? How can we learn from this? And so with microorganisms help by becoming cell models. For years, microorganisms are used as a model for scientists to study. As they study these microbial cells, they can learn more about cells in general. I mean, we know now, and at this point in our lives, we have all kinds of advances in medicine that, people did not have even just 20, 30 years ago. And but definitely way back, beyond that. But it's because of science and studying and being able to use these types of cells to learn how we can, interact with them. We can provide medicine for things, so that's what science is, and the study of not just medicine, but in general, these microorganisms help to play that role. I can't remember if it's actually, I think it is page 5. It might be a slightly different page in your all's book. But there's a table in your all's textbook that and I'm still going to talk about it in the PowerPoint, but it does talk about different industries that microorganisms are used in. So such as the food industry, alcoholic beverages, chemicals, antibiotics. We'll talk about that again in just a second. This picture just provides an example, as I mentioned before, that microbes are very much involved in different elemental cycles. This uses the example of nitrogen. Nitrogen is in our atmosphere. We don't typically breathe in nitrogen like we do oxygen. Nitrogen is something that human beings have to get from eating proteins. But it is important for our atmosphere and for other things in our world to take in this nitrogen. So nitrogen is important for our atmosphere. So it needs to be in the air, and microbes help to play the role in that. As you can see, as it shows, the nitrogen fixing bacteria is helping to provide the nutrient there for the soil, and then nitrogen can return to air, and it plays that role in that cycle. As I mentioned, about the table that's in your textbook, it's here as well. Microbes are used in many industries, such as food. If you guys remember, just one that you guys are probably pretty familiar with, yeast. Yeast is a fungi. Yeast is an example of a fungi. Sometimes people think yeast infection, but again, that's a totally different kind of yeast that causes a yeast infection. But there's a certain kind of yeast that is used to help bread rise. So it's used in making dough and making, bread. Another example with food would be, we think of the term mold as something that's bad, because mold is something that grows on rotting food in our refrigerator. Different type of mold. There's another type of mold that is used to give the flavor to certain cheeses, like blue cheese. Some people don't like blue cheese. I myself love blue cheese, but that's exactly what it looks like too. It looks like mold growing on cheese, but like I said, it's a totally different kind of mold. This is a mold that's good for us, that can be eaten, and it is just what's giving it that flavor. Swiss cheese, all the holes in Swiss cheese is actually caused by bacteria. So again, microbes are used in many different ways in the food industry, beverages as well. We think about wine and beer and things, alcoholic beverages, the fermentation process and the aging process, as it sits, it becomes alcoholic as it, again, as these beverages are, getting that, what it needs to become that beverage. It's microorganisms that are helping in that fermentation process. Different chemical industries, antibiotic industries. Just as kind of a fun fact, we'll bring this up when I talk about chapter 9. Chapter 9 is what covers the antimicrobial. I mentioned chapter 9 earlier with fungi. It's actually chapter 5 that we're going to cover fungi. But antibiotics, like I said, I'll mention this again. Alexander Fleming is the name of the scientist who discovered penicillin, a very common antibiotic that we're probably very familiar with. Some people cannot take penicillin because they're allergic to it, but it's a very, very common antibiotic. It was actually accidentally discovered by Alexander Fleming. What he had done is he had left, bacteria growing on a petri dish. And what happened was mold happened to get on that petri dish by accident. And what he noticed was around that mold, the bacteria started disappearing. It started dying off. And he thought, wait a minute. And that is how he eventually discovered penicillin. So he studied it some more and was able to discover that mold is what actually caused the killing off of that bacteria. So it's again, different microorganisms that can even help get rid of other microorganisms. And also used in genetic engineering. In genetic engineering, what happens is genes. So again, we talked a little bit earlier about that DNA, right? DNA in our cells, that's that genetic information. Genes from one organism are then inserted into a bacteria or a yeast cell And the cell that receives the new gene is then capable of producing gene products coded for that gene. So they're able to, again, as I mentioned earlier, microbes are used for science and used for studying, and we're able to see how these, how taking gene information, so genetic information, from one cell and taking it and putting it into another cell, how we can then create new products from that. The use of living organisms or their derivatives to make or modify useful products. This process itself is called biotechnology. So microbes are used in that process as well. So, okay, so I do have this. Good. So some of the first microorganisms on Earth, fossils of primitive microorganisms date back to about 3.5 billion years ago. Candidates for the first microorganisms on Earth are what's called archaea and cyanobacteria. Those are some of the first microorganisms that we discovered. Infectious diseases of humans and animals have existed for as long as humans and animals have existed. Again, it's science that we have discovered all these advances in medicine, and what we know now, we did not know a very, very long time ago. We had no idea that it was these germs or pathogens that was causing people to get sick. And we'll talk about one important pioneer in microbiology and how he was able to discover that. So some of the earliest known account of pestilence occurred in Egypt in about 3180 BC. So it's just talking about how we were able to discover that back we can kind of date these fossils and how far back they were, but we're seeing that microorganisms have been around forever. Microorganisms have been around longer than humans have been around. So we're seeing that, again, they've always been around, but now we're able to study them a little bit further and see that a lot of them, again, are really good for us and providing a lot of helpful things for our atmosphere, for now our food and medicine industry. So we're, again, discovering a lot about these microorganisms. So let's talk about some of those that first discovered some of these microorganisms, but especially in the study of microbiology, but especially as we talk a little bit more about medicine. We'll get into that with the last pioneer here. We're going to talk about three people that were really, really important in the science of microbiology. The first one here, Anton van Leeuwenhoek. As you can see, Anton van Leeuwenhoek was around from 1632 to 1723. That was a really long time ago. So compared to now, we obviously know a lot more now, but back then they didn't know that these microorganisms even existed. So that's why he is called the father of microbiology. He's not a trained scientist. So he's not, again, he's not categorized as a scientist. He doesn't fall into that category, but he's more of an inventor, really. He invented a simple single lens microscope. And again, we're going to talk a little bit about microscopes in the next chapter, but he was able to develop something that would allow him to see smaller objects, that he wouldn't be able to see with the naked eye. He was able to observe what he called animalcules. He called them animalcules because he saw something moving around and living at this small, tiny life form. So he associated with animals, but he knew that they were much smaller. So what he was actually looking at was bacteria and protozoa, which are a little bit larger than viruses, and we'll talk about that. So he didn't have anything that would be able to see viruses just yet, but he was able to take a look at a microscope. Again, most microorganisms, or not most, microorganisms cannot be seen just with the naked eye. So this individual, Anton van Leeuwenhoek, was able to develop an instrument, again, which we know now as a microscope, but he developed this instrument that allowed us to see that. And since then, microscopes have been, have come a long way and have, are much more advanced now. But then it was at least going to be able to be something that allowed him to see these smaller organisms. So really, he's the first person who was able to see microorganisms that he was able with this instrument here. So that's why he's the father of microbiology, because he was the first to come up with a microscope and therefore the first to view microorganisms. So Anton van Leeuwenhoek is very important for that reason. And then there's Louis Pasteur. Pasteur, again, this is way past Anton van Leeuwenhoek. So microscopes have been around, probably advanced a little bit further, but you're seeing 1822 to 1895. So as science has come a long way now, science has come a little ways since Anton van Leeuwenhoek at this point, but not quite. But Louis Pasteur is going to help with a little bit of this. Louis Pasteur was a French chemist. He made numerous contributions to microbiology. One would be that he investigated different fermentation products. I had talked to you guys about how fermentation is what helps to age things like wine and beer, and fermentation is used for lots of different things. But he was able to help investigate that and learn more about that. The biggest thing about Louis Pasteur that I think is really important to remember, because you can see his name is in this process here, he actually helped to develop the pasteurization process. The pasteurization process, which we know now, things like milk and cheeses go through pasteurization because it helps to remove some of the bacteria that's in those products, and it helps to keep them a little bit longer. So that's because of him. Louis Pasteur helped to develop that pasteurization process. He was able to discover that life forms would exist without oxygen. So there are some life forms that need oxygen in order to survive, but there are some that can live without oxygen, and they're referred to as anaerobes. So it's a type of bacteria that we'll talk about when we cover our bacterial chapter, but he was the first to discover that. He also helped to develop several different vaccines, including rabies and anthrax vaccines. So at this point, we've come far in medicine, and we're able to then develop different vaccines, meaning we're helping to keep people from getting these diseases at this point. So he's helping to develop that. So Louis Pasteur, really important scientist who helped make numerous contributions to microbiology. All right, and then Robert Koch. Robert Koch, a little bit past Louis Pasteur's time. He's here from 1843 to 1910. So just again, we've come a long way, but Robert Koch is really going to be really important in microbiology, especially for nurses. And here's why: because this individual actually helped to understand that there are pathogens. That pathogens, again, we commonly refer to those as germs, but he's able to discover that. So that's why Robert Koch is going to be really important here. So he's a German physician who made also numerous contributions to microbiology, but his biggest thing is going to be the germ theory of disease. Now, at this point in our lives where we stand, we know that one microorganism, for example, influenza, again, we know the specific microorganism that causes the flu virus. We know the specific microorganism that causes a cold. We know the specific microorganism that causes chickenpox. But at this point, Robert Koch is helping to study that. So he made significant contributions to that germ theory. He also discovered that Bacillus anthracis, which is a type of bacteria, produces spores, meaning that that's how it's going to reproduce and how it's going to spread. We'll talk about that a little bit further when we get into our bacteria. He developed methods of fixing and staining bacteria. We'll talk about staining when we talk about bacteria too, but bacteria, difficult sometimes to see on a microscope because a lot of bacterial cells are clear. So if you shine a light through it and look through a lens, it's going to be hard to see because all you're going to see is a light. But Robert Koch helped to develop a process where we can take a stain and help to adhere it to a slide. That's what fixing means that it stays still. We can I'll talk to you guys about this in chapter 2, but it helps to keep it in place, and the stain helps to darken that bacteria so we can see it a little bit better. So he helped to develop that. And he also helped to develop different ways to cultivate bacteria. But let's talk about that germ theory. That germ theory is really, really important, especially, like I said, in healthcare and nursing. So here's what I mean by this. Robert Koch developed what's called these postulates, which is kind of like his hypothesis. He says that a particular microbe must be found in all cases of a disease and must not be present in healthy humans or animals. Again, today we know that to be true, that if person A and B both are sick with the flu, well, they probably have the virus that causes the flu within their body. They have that particular strain, whereas person C and D are healthy as a horse. they don't have the flu virus. So they're, that particular microbe is not found in them. The microbe can then be isolated from the diseased animal or human. So if we take, say, a blood sample or a saliva from person A who has the flu virus, we can isolate, from that diseased person, and we can grow it in a laboratory. So if we can grow that particular disease in a laboratory, but then if we then take it from the laboratory, take that same disease, and take it and inoculate it into a healthy human, if that healthy human gets sick, then it means that's what causes that disease. So that's what it means there, number 3. The same disease must be produced when microbes from that pure culture are inoculated into a healthy, susceptible laboratory animal. If that laboratory animal gets sick, then again, we're able to kind of prove this germ theory. We're proving Koch's postulates. So the same microbe must be recovered from the experimentally grown again in pure culture. That's what science is. Science is experiment after experiment after experiment. You have to prove that that's what's causing that in order to make it true. You have to make sure that that's what's causing that particular infection. So this just gives you a little bit of a visualization. So here, this individual, this particular mouse is sick. So if we were able to take that microorganism from that sick mouse, grow it in a laboratory, then isolate it in this culture here, take it from that culture and inject it into a healthy mouse. If that healthy mouse again then gets sick, we can then take it from that sick mouse and we can do the same process over and over again. And that proves that it's that particular microbe that causes that particular infection. And again, science has come a whole long way from this, because again, we've been able to see that, okay, well, there's the flu, but now we can do the same thing with, hepatitis. We can do the same thing with measles and mumps. We could do the same thing with all these other, infectious diseases that are caused by microorganisms. We're able to see that now. We're able to discover that and see that and study it, and then eventually study what gets rid of it, and develop vaccines from it. And that's later. But right now, we're just discovering that it's these particular pathogens that are causing these particular diseases. So if an organism fulfills Koch's postulates, it has been proven to be the cause of that particular infectious disease. Koch's postulates help prove the germ theory of disease. Robert Koch gave a tremendous boost to the development of microbiology by stressing this laboratory culture and that identification of these different microorganisms. And yes, there are sometimes circumstances that do exist where Koch's postulates cannot be fulfilled, meaning that maybe we take a microbe from a sick animal and it doesn't make the next animal sick, or we can't grow it correctly in pure culture. And sometimes that it cannot be fulfilled, but that's where we just keep studying. And that, guys, is the end of chapter 1. So just to finish up with this, just as typically this chapter ends with careers in microbiology, and you guys have already picked a career in not in microbiology, but in general in nursing. And why microbiology is going to be important for you all is, again, because you guys are going to see the ones that cause these infectious diseases in your patients. You guys are going to see and understand that, as we talk about, viruses and bacteria and fungi, those particular organisms that are pathogenic when they make your patient sick, we're going to talk about what we can do to hopefully prevent that, treat that, how we diagnose it. So we're going to talk as we go later into the course, we'll talk about ways that we can try to get rid of these infectious diseases, how hospitals and healthcare facilities are making sure that we're keeping these infectious diseases away from these patients. So that's what we're going to take a look at. So you guys will see that as we go throughout this chapter, throughout this book and throughout this course, that microbiology is going to be very important for nurses. And that's our introduction into microbiology. All right, guys, so that's chapter 1. If you guys have any questions, let me know.